Okay, so in this session we're going to spend some time addressing the way in which we can monitor the blood lactate response to exercise and use that as an indication of what is referred to as the aerobic capacity. So in previous sessions we've identified key endurance parameters. We've identified, for example, VO2 max and have referred to that as an indication of maximal aerobic power. We've identified submaximal responses such as running economy and identified how important that is to endurance. But one of the things that we recognize is that there is a point during incremental exercise and there is a point in terms of systems physiology that represents the boundary between predominantly aerobic energy and predominantly anaerobic energy. And that is what is referred to as the lactate turn point. And if we can identify this point, this transition place, we can then start to evaluate endurance capacity, and particularly this idea of an aerobic capacity. And we can start to monitor this both in athletic groups and also in groups who are perhaps more clinically orientated. So this is a fundamental component of endurance physiology. To do this, we're going to go through the following areas. We first of all need to explore the concept of the aerobic capacity and, importantly, the taxonomy, the terminology that goes with it. And this actually is a very, very complicated component. It's not complicated in terms of the underlying physiology to a certain degree, but it is complicated because there is an awful lot of terminology that exists that just seems to overlap and is misconstrued and is almost misdirecting us. So we need to clear that up. Then we're going to explore the role of lactate data in a physiological setting. Why do we assess the blood lactate response to exercise? Why is this considered to be important? Remember, one of the key concepts within assessing athletes is we only really use these tests if they are considered to be valid and also if they are reliable. And so that second point really drifts into the third point, which is, well, what is the rationale for assessing blood lactate data? Why is it important or considered important in a clinical population group? And why do we consider it to be of importance in an athletic population group? And then finally, the big part, I suppose, of this podcast will be how we're going to go about identifying this aerobic capacity, this transition between predominantly aerobic and predominantly anaerobic metabolism. And as there is an array of terminology, there is also an array of tests or protocols that have been developed. And what we've got to do is go through these and again fall back on that notion of, is the protocol valid? Is it reliable? If it ticks both of those, then it works. And we're going to spend some time addressing those as we go through.